period ఎవరైనా క్రైమ్ చేస్తే ఆ అక్యూజ్డ్ పర్సన్ తీసుకొచ్చి ఒక ఫోరం ముందు నిలబెట్టేవారు. నిలబెట్టేసేసి దే యూస్ టు మేక్ ఆర్గ్యుమెంట్స్ అండ్ ఫైనల్లీ అతను చేసిన తప్ప ఒప్ప అని చెప్పి నిర్ణయించేవారు. The code has come from that forum. It turned to the forensic science. And it's if you see, if you go to the back of the history of the forensic science, this person, Sir Arthur Conan Doyle, is the he has given many inputs to forensic science. He is the author of that Sherlock Holmes novel. It's a very good detective novel given by this person, written by this person. And he mentioned many innovative detective ideas in that. Forensic science has emerged based on these ideas, his ideas. So, the serology, document analysis, firearm identification, crime scene observation, and evidence analysis. These are all taken from his novel, and many movies also, detective movies also have come based on his novels. Next. And Archimedes, have you heard about the Eureka concept? Eureka? Anybody aware of this? Forensic attempt, see that goes back to the 287 to 212 BC. Archimedes was a minister at king, and one day ఒకసారి కింగ్ ఏం చేస్తారంటే ఆ క్రౌన్ ఆర్డర్ చేస్తాడు, గోల్డ్ క్రౌన్ ఆర్డర్ చేస్తాడు. ఆర్డర్ చేసినప్పుడు గోల్డ్ స్మిత్ ప్రిపేర్ చేసి ఇచ్చిన తర్వాత he has a doubt whether it is pure or adulterated. Then without damaging to the crown, he asked Archimedes to find out whether it is a pure gold or adulterated. Then he was thinking how damaging the crown. One day he was taking a bath in the tub. Some amount of water dislocated. So they immediately got an idea. So ఆయన ఎంత వెయిట్ ఉన్నాడో అంత వెయిట్ ఉన్నటువంటి వాటర్ అనేది డిస్లోకేట్ అయింది. Then he used both pure and adulterated gold, and he finally concluded that that gold, that the crown was adulterated, and immediately ran from the tub to the king. Eureka అలా బట్టలు లేకుండానే పరిగెత్తుకుంటూ వెళ్ళాడు అనమాట. దట్స్ అ కాన్సెప్ట్, యురేకా కాన్సెప్ట్. యురేకా మీన్స్ ఐ ఫౌండ్ అవుట్. సో దట్స్ ద ఫస్ట్ అటెంప్ట్ ఇన్ forensic science. Next. So this is the fingerprinting technology. Nowadays we are using the thumb. Wherever you go, we are using a thumb impression. And first Chinese used thumbs, thumb impressions, and they analyzed. That was the first attempt made by the Chinese in the 1700s. Next. So these are the various branches. And father of forensic science goes to the Bernard Spilsbury. He invented so many ideas, theories in forensic science. And the father of modern forensic science is Edmond Locard. And there are different. The father of forensic botany, Arthur Koehler. Next. And he's a person called Bryant. Wagner Bryant is a great paleontologist who worked hard and solved many crimes by using the paleontology. So he is said to be father of forensic paleontology. Next. So there are 7 basic principles entire forensic paleontology works on. That's the first one is the law of individuality. Every object in the crime scene has individuality. You cannot make a comparison. Each one has its own identity. Principle of exchange. Next slide. Principle of exchange. For example, I am here. If I touch this podium, I am contributing something from me to this. Finally, come out with the conclusions. Next. These are the various areas of forensic science. Forensic science is not an independent science. It has a multidisciplinary. It connects with the many disciplines. That's anatomy, maths, linguistics, social studies, biology, physics, technology, whatnot. Every subject is connected to the forensic science. Next. And here is a forensic botany, particularly forensic botany. We go with the anatomy, we use anatomy, morphology. How many of you here are botany students? Very few. Yeah, very good, very good. So you all know what's the morphology. Morphology. The basic appearance of the plant is called as a morphology. The study of the external features of the plant is called as a morphology. Okay, we use that also sometimes. And anatomy, internal structure, and palynology. Okay, and limnology, the diatoms and other microorganisms, phytoplankton, zooplanktons, they're part and parcel of the lakes. They're also used. Next. And next, coming to the forensic palynology, application of the palynology to solve the crime or legal aspects is called as a forensic palynology. Palynology is nothing but it is the study of the spore and pollen grains. Apart from that, all other microorganisms which range from the 5 micrometers to the 100 micrometers. They're very, very tiny particles. Very tiny particles. They are used here, and this is the case first solved by using the forensic palynology. In the year of 1959, Sweden, a lady was murdered and dumped somewhere. Nobody knows who is she, because she murdered elsewhere and body was dumped some other place. Nobody knows. And a few years back, one person kept the lady body in a suitcase and left it at Medipatnam bus stop. Still, till today, the case was not solved. Like this also, almost they want to close it. But finally, they have given to the palynologists by observing that they have collected some dirt particles from the clothes of this lady, and they have compared with the surroundings. That pollen is not matching with the surroundings. This is ambrosia pollen, which is not part of that, and which goes mainly in one particular locality of that Sweden. And they started searching there, and there they find our relatives, and finally they solve this crime. Next. See, this is a study of pollen and spores. Left side you are seeing they are the beautiful pollen grains, and right side they are the spores. Next. So difference between the spore and pollen. Spore is produced by the lower plants from Thallophyta, Bryophyta up to Pteridophyta. They produce the pollen spores, and whereas Spermatophyta, gymnosperms and angiosperms produce the pollen grains. Spore has got a wide ecological niche. They are omnipresent. Spores are omnipresent, and they get wherever suitable media they germinate. For example, if I place bread here, after next day, what happens? What happens? There is a growth of mycelium. Mycelium. From where is it has come? The mycelium has come because of the spores present here. If you place the bread in any cell or in one box and see after 2 days or 3 days, again you can see the growth of the mycelia there. That means the spores are present everywhere. Wherever they get a suitable media, there they germinate. But whereas pollen cannot germinate like that, and it has very, very restriction. It germinates only on the stigmatic surface. Stigmatic surface. So this is the pollen grain. Generally, anatomist or a cytologist looks for inner contents of the pollen grain. Embryologist looks for the germination aspects of the pollen grain, because pollen has to germinate on the stigmatic surface and pollen tube emerge and reach the ovule. There it discharge the 2 sperm cell, 1 sperm cell fused with the egg cell and other sperm cell fused with the secondary nucleus. That's a double fertilization. 2 fertilization takes place. Okay. So those germination aspects dealt by the embryologist, whereas a palynologist doesn't look for all these, only look for the outermost layer that is called as an exile. See that outermost ornamented structure you are seeing, no? That is the egg side. Only the clues, the morphological aspects, the designs present there, they are only helps in the identification part. And we use that part, and recently we have come out with a new concept, that is Paragam it, and we got granted patent also, that we use those designs in the textile industry for formation of various designs. We named it as Paragam. Next. See here, the germination of the pollen grains on the stigmatic surface. So there is a flower, for example, you see, this is a flower. This is a flower, Hibiscus rosa sinensis, which is exposed to the open environment. Okay, and this part is stigma. Okay, once it is open to the environment, not only hibiscus pollen, it also receives the Tacitus indica or Rosa indica or Chrysanthemum indicum, or else many other pollen grains also come and land over here. It has to recognize its own pollen and allow for its germination. It won't allow other pollen to germinate on the stigmatic surface. For that reason, each and every plant produces a very unique type of pollen grains. That uniqueness is in the form of pollen units, polarity, shape and size, symmetry, apertures, and extent stratification. Very beautiful extent stratifications. See this flower you are seeing, you cannot see the pollen grains unless otherwise you have a microscope. But those teeny structures are playing a great role. Okay, they are giving many clues to solve the crime also. See here, Anopheles plants, generally the pollination is nothing but reaching of the pollen grain from anther lobes to the stigmatic surface. That's the pollination. Pollination done by many ways. So for that, plants always give some advertisements. You know all advertisements. Why advertisements? Enduku advertisements enduku istaru? Advertisements enduku istaru? Enduku? To attract. Here also, plants also go for advertisements. Floral advertisements in the form of colored petals, in the form of scented petals, in the form of various patterns. To that, with that they attract the pollen vectors and they get a pollination. Okay, Anopheles plants generally they shed a huge amount of pollen grains. See there, you see that gymnosperm pollen they are releasing like a cloud. That's called pollen rain. Because air has to carry that. Okay, next. See here, this is a Zoophilus or Entomophilus plants. They are very sticky, so that the pollen grains are attached to the body parts of the bee, and it carries that pollen. So these are various number of pollen grains released from the anther lobes, huge amounts. Each plant has. So what forensic pathology can do? Determine the travel history of the items or person. Morning I have come from the Hyderabad to LRT. Okay, if you analyze me, the pollen exactly, you can get of the Hyderabad flora between the Nasapur plants and also LRT. So that you can determine travel history from where he has come. ఎక్కడి నుంచి వచ్చాడు అని చెప్పేసి మనం అనలైజ్ చేయొచ్చు, ఫైండ్ అవుట్ చేయొచ్చు. The pollen which are attached to the body parts of the human beings. That's the greatness of this. And next one is provide information on the environment an item has come from or origin. A environment lo undi, whether the person has come from the xeric environment, xerophytic environment, or wetland environment, or hydrophytic environment, or mesophytic environment, or from forest, or from agriculture fields. You can identify. If the person has come from the agriculture fields, అగ్రికల్చర్ వరి గోధుమ లేకపోతే అటువంటి ప్లాంట్స్ ఏదైతే ఉన్నాయో ఆ పోలన్ గ్రెయిన్స్ అన్నీ కూడా బాడీ పార్ట్స్ మీద దొరుకుతాయి, షర్ట్స్ మీద కానీ హెయిర్ మీద కానీ ఉంటాయి. So by analyzing that, we can come to the conclusion that the person has come from the agriculture field. Right? Interesting. Getting bored. If you feel bored, you just raise your hand. Okay. And next, determine the body was moved. Body was moved. ఆ బాడీని ఇక్కడ నుంచి వేరే చోట పడేస్తాం అనుకోండి, ఇక్కడ ఉన్న పోలెన్ వేరు ఉంటుంది, అక్కడ ఉన్న పోలెన్ వేరు ఉంటుంది. విత్ దిస్ ఫస్ట్ కేస్ వాస్ సాల్వ్డ్. అండ్ నెక్స్ట్ determine the season in which crime was occurred, committed. సీజన్ కూడా మనం ఫైండ్ అవుట్ చేయొచ్చు. ఎందుకంటే మెజారిటీ ఆఫ్ ద హర్బ్స్ కమ్స్ టు ఫ్లవరింగ్ ఇన్ ద మంత్ ఆఫ్ during the months of September to December. Majority of the trees comes to flowering during the months of January to May. Okay, so month wise also we can find out. Right. So what makes a pollen so useful? They are so microscopic. ఎవడన్నా క్రైమ్ చేద్దామని కూడా వచ్చాడు అనుకోండి, వాడు చేస్తారు అంత ప్రికాషన్స్ తీసుకుంటాడు, బట్ he cannot see the pollen and spore which are present here. Without noticing them, he leaves the traces and he'll go out. Definitely, that will become evidence, and finally, that person will be caught. Okay, that's the beauty with the palynology. Microscopic generally goes unnoticed, and the suspect picks up evidence or his or her presence at a crime scene without realizing it, therefore without taking precautions. Variable morphology. Each pollen has different shape and size and all these, so that we can make out the difference. And durability, the exine I told you, exine is made up of a sporopollen. That's a very hard, tough, rigid layer which cannot be destroyed by the biological, physical, and chemical alterations. That can be for years. If that is preserved properly, years long, that can be preserved, and after 10 years, 100 years also, you can take out and you can find out what type of pollen it is. Okay, next. So this is the methodology generally we use: identification, preservation, analysis, documentation, presentation. So collection of forensic samples requires the control and evidence samples. Both are required. Control sample means, for example, there is a dead body surrounding within the radius of the 10 centimeters or 15 centimeters, we have to collect some samples. Those are called as the control samples, and whatever the samples we collect from the dead body, they are called as evidence samples. Okay, so we can make a comparison. Both are there, both are matching. That means the crime has occurred there itself. If they are not matching, that means elsewhere it was occurred and the body was shifted. That we can confirm. Okay. Next, these are the various kits we use. Next. This is the pinch method generally for control samples. 5 to 10 grams of material. Generally sticky tape method we use, and we collect the samples, both control and as well as evidence samples, and we have to seal it and put it in the custody. Next. So generally, whenever a crime scene is there, crime occurred, forensic scientists ask for palynologists to collect the samples, both evidence and control samples. We have to collect these, we have to look for these. Hair, dust and sediments, back. Dust and sediments, clothes, shoes, industrial parts, vehicle, vehicles, furniture and all these. From these parts we have to collect the samples. See here, hair. Every human being hair is different, looks different. Somebody's hair is straight, somebody's hair is curly, somebody use oil, coconut oil, or somebody use castor oil. It varies. So based on that, it holds many pollen. So hair is a good source of the pollen. Okay. So while walking, you apply coconut oil and you have a curly hair. You're walking. Just you walk for 10 minutes. you can collect the many 100 to 200 pollen grains from your hair. That's the beauty of this. Okay, next. Even clothes also. People wear different types of clothes in different environments of the Indian system. Okay, so each type of clothing attracts pollen varily. Silk, cotton, hairy cotton, etc., etc., etc. We use different types of clothes. They also hold the pollen as per the cloth material. Next. See these rug fibers, that's a rug generally used during the winter season. Molds, pet dander, bacteria, skin cells, pollen, dirt, dust mites, all are part of that. If you analyze the rug, you can extract all these. Okay, and they gives you beautiful clothes, even shoes also while walking. And this is next. Honey samples. See, person is coming from the Kashmir to Kanyakumari, traveling. Suddenly, he killed between, and nobody knows from where he has come. He is holding a sample of honey. Sample of honey. If you analyze that sample, if you find a Crocus sativus, what is that Crocus sativus? Anybody? What is students? Crocus sativus. Saffron, కుంకుమ పువ్వు అంటారు కదా, తెలుసా? కుంకుమ పువ్వు ప్రెగ్నెంట్గా ఉన్నప్పుడు పాలలో వేస్తారు, అబ్బాయి ఎర్రగా ఉండాలని చెప్పేసి. అతను ఎక్కువ వేయకండి, చచ్చి ఊరుకుంటాడు. That's a poisonous in higher doses, higher levels. Okay, so that's the Crocus sativus that only grows in Kashmir or rhododendrons. That gives you geographical print. Okay, so that analysis gives you. And if you have a small amount of honey, 1 gram or 10 grams of honey, I can tell you from which geographical locality it has come, from which plant it has come, how many plants bees were visited, everything can be revealed out just based on studying of analysis of the honey sample. That's called as another application of the technology, melissopil knowledge. That's the beauty with this. Okay, next. And imported dry fruits determine the country of origin. Dry fruits, different places they harvest and they import and they give. So, which country they harvested or imported from, we come to know. Next. And coffee, tea, yeah. Here, there you see the most expensive coffee in the world is Kopi Luwak. Kopi Luwak. Kela tayar chesaro telusa? This is very, very expensive coffee. Generally, in Tirupati and there one cat that's called palm civet. Okay, Asian palm civet. పురుగు పిల్లి అంటారు. ఆ పురుగు పిల్లి ఏం చేస్తుందంటే బీన్స్ తింటుంది, కాఫీ బీన్స్ తింటుంది. అరకు కాఫీ చాలా ఫేమస్, మొత్తం వరల్డ్ ఫేమస్. ఎందుకు అంత ఫేమస్ అయిందంటే అది తిన్న తర్వాత ఏమవుతుందంటే ఆ beans అనేవి they don't get a digestion inside the intestine and that cat excretes. అవి కలెక్ట్ చేసేస్తారు. కలెక్ట్ చేసినప్పుడు ఆ లోపల ఉన్నటువంటి ఎంజైమ్స్ తోటి అదంతా కూడా డైజెస్ట్ అయిపోయేసి దట్ గివ్స్ ఎ గుడ్ ఫ్లేవర్ టు దట్ బీన్స్. దాంతో తయారు చేస్తారు. దట్స్ ఓల్డ్ కాస్ట్లీయెస్ట్ కాఫీ. ఓకే, అరకు కాఫీ. అండ్ వెదర్ దిస్ ఈజ్ ఎగ్జాక్ట్లీ ఫ్రమ్ దట్ కాఫీ ఆర్ నాట్, అరకు ప్లేస్ ఆర్ నాట్, or in some other place or not, we can confirm with the well knowledge. Even pollen grains also cannot be digested. I told you, the exile is very tough. Biological system cannot be destroyed. Okay, so the attached pollen to these, as these we can find out and we can come to the next. Even currency notes also, coins also sometimes they hold, rarely. Least case, you can go with this evidence. Next. So air filters, all your vehicles moves different places. Okay, every time they are moving, the air is filtered, and while filtering, it also filters all these dust particles, pollen grains, and all these. Once you take out that air filter and you analyze it, you'll come to know the vehicle has moved from so and so places, from so and so geographical areas. It has moved. That we can determine. Next. And what is this? The left side? Opium. What is the common name? Opium? గసగసాలు మీ కిచెన్లో ఉంటుంది గసగసాలు బట్ దట్స్ నాట్ గసగసాలు సీడ్స్ యాక్చువల్లీ దే ఆర్ నాట్ ఎగ్జాక్ట్లీ దిస్ బట్ ద లేటెక్స్ ఆఫ్ దిస్ క్యాప్సూల్ see the capsule, inside that capsule you get that గసగసాలు but opium you get from that latex ooze out from that capsule. They tap that capsule, latex వస్తుంది దాన్ని డ్రై చేసే you get opium. Opium is very dangerous and highly next ganzai. That's go back. That's ganzai. Nowadays is very problematic. So if you analyze cannabis sativa that looks like a gongura, hibiscus cannabinus. Gongura lane undi kada, hibiscus cannabinus. That's why this is called as a cannabis sativa. Okay. If you analyze the pollen attached to this, you can say clearly, you can find out clearly the geographical source, whether it is from Andhra or Odisha or some other nearby places. If you go to the north, UP particularly, there is a common weed, ekkada patte akkada perugutundi, ganjai, but here it is prohibited. Okay, next. And this is the arms, one beauty with this. If you coat with the pollen, each bullet, each batch of the bullets, once you trigger it, the bullet comes out. With the high temperatures also, the bullets are not damaged because physical parameters also cannot destroy. The outermost layer is made up of sporopollen that withstands the temperatures. So we can know which bullet is used, from which arm it is used. Okay, those things also can be revealed now. Next. Can you switch on this one? Extraction of pollen and spore samples. Generally, we have to extract. That's very lot of patience we require because these are very tiny particles. Can you see the pollen here? You cannot see. You cannot see. So if I want to extract the pollen, I need a lot of patience, observation, okay, and cleanliness also, so that there won't be any contamination. Only the pollen of that particular locality or particular body parts or particular sediments we have extracted from that can be extracted. Next. So this is the analysis we do. I'm sorry, I will wind up within 10 minutes. Next. So this is the latest technologies from 1930s to 2019. If you see, there's a lot of development in the science. Okay, light microscopy too. Now we have orthogonal analysis. At a time we can analyze so many samples. Next. So this is one example. Last time also one person. Forensic science we have in our university. Okay, there one case has come. Hussain Sagar lo okatani padesaru. You can do things. Left side you are seeing the person attempted suicide. అందులో దూకాడు చచ్చిపోయాడు. Right side the person killed somewhere else and dumped there. ఎలా అనలైజ్ చేస్తాము, ఎలా ఫైండ్ అవుట్ చేస్తాము సూసైడ్ అటెంప్టా or murder attempt అని చెప్పేసి. See, the lungs, immediately the person attempts suicide, he swallows water, engulf water. Water మింగుతూ ఉన్నప్పుడు ఏమైందంటే that goes into the lungs. Along with that water, many diatoms, other microorganisms also enters into the body parts and with the intestine and lungs also. Okay. After some period, all your inner parts also get contaminated with this. They destroy that because already they enter into your body. But whereas there, they won't enter into the lungs because already it's a dead body. You're dumped there, and they are clean. They are free from the microorganisms. Okay. Next. And I would like to present the two cases, important cases. One is case one, that's a Texas matter. In Texas, one person was killed. Only that skeleton part was available with that shirt and t-shirt and pant and shoes. Nothing else. And it was given to the biologist. Okay. And finally, they found one algus and Shapatia, these two pollen grains there, and they are not part of that Texas. They grow only in that West Texas, and then the search moved to the from North Texas to the West Texas, and finally they changed this. Next, and this is another important case study. Anybody here follow Christianity? Yeah. Christ was died after his crucifixion. He was buried in that tomb, and then he was wrapped with that cloth. Okay, wrapped with that cloth. That's called as a shroud. That is displayed in Europe now. That is in Turin. That's called as a shroud of Turin. Okay, and after third day he resurrected. After third day he resurrected by leaving the cloth there itself. And that cloth was collected and displayed in Turin. You know, that religion was moved from there to Europe, so that's why they have displayed there. And many of them raise a question about that cloth, whether it is real original cloth when Jesus was used, or that dead body was wrapped in that or not. Then this case was given to many scientists, forensic scientists worked on that. They all proved this is from Israel, that region, that's the original region. Even finally it was given to the Max Frey, is a person, right extreme person. He was also worked on that. That person also worked on that. And he got nearly 58 different pollen taxa. 58 different taxa. Pollen types. 58 pollen types he recorded from that cloth. And interestingly, these four key types pollen grains, zygophyllum and caparis, other pollen grains, these are all xeric environment, and most of them are from the habitats, xeric habitats of the Israel and Turkey. Okay, where the crucifixion was occurred. Finally, they proved it. Some pollen also trapped there, and they are showing European pollen because of while displaying that cloth there, they trapped. But remaining all important pollen trapped there, they are from this region. So they also concluded, biologists also concluded that this is the exact cloth trapped to the body of Christ. Next. So opportunities in forensic science and careers. Next. So there are so many various opportunities. You can work as many branches are there. Forensic science, many branches are there. As per your interest, any field, whether it is a BSc, BA, or any degree, you can enter into the PG, or else from intermediate you can enter into the BSc. Next. There are different universities, colleges offering. This Usman University is offering forensic science, MSc, and Loknaik Jayaprakash Narayan. That's one. University of Madras is also offering. Rajasthan, Tutoji, Maharashtra Nagpur University, and all these Banaras Hindu University is also offering. Institute of Forensic Science, Mumbai, Government Institute of Forensic Science, Punjab University, all these universities are offering this course. Okay. Back. See, this is the National Forensic Science University located in Gujarat. Okay, and it is opening many branches throughout India. They want to open. Presently, they have started their branches here, almost 16 places, in 16 states. Next, and last year it was occurred All India Forensic Science Summit 2025. Shri Amit Shah was the chief guest, and he announced that time. Every state they want to establish one university or one center, one institution, completely work on the forensic science because the crime rate has been increased. To address this challenge, they want to start, they want to initiate. Next. 2 minutes, within 2 minutes. So there are every year there are many opportunities. They are giving vacancies, job vacancies. Next. So NFSU, National Forensic Science University Recruitment 2026. Recently they have announced. Next. So Delhi Forensic Science Lab also recruited. And to get ready for, if you are interested, you can join either for BSc or MSc Forensic Science and have a good career. Thank you all. Any doubts? So looking hungry. Yeah, thank you, Anita.
Today, it's a relation between